Okay, good afternoon, everybody. You guys have had a great day since I saw you yesterday. Could I just get a quick confirmation that you guys are seeing the PowerPoint slides for muscle tissue? Thanks, Deacon. Okay, cool. So looks like a small class today, but maybe we'll get some people rolling in as we go through this. So this will be our last chapter in Unit 2, Chapter 10 on muscle as a tissue. And so we have some options for how we utilize Thursday's class and next Tuesday's class, which will be our last two meetings before you take the actual lecture exam, which is next Wednesday. So I'll talk about that more at the end. For now, let's just get started with the Chapter 10 content. So we've been learning about these specific muscles in our body during lab, but this is going to be focusing on the actual structure and composition of muscle as a tissue. So trying to look at the individual structures that make up this particular tissue and how that relates to the various functions that muscles perform for our body. So we have lots of topics to touch on in this PowerPoint. There are a few topics from the chapter that we are not discussing explicitly in this PowerPoint or in lecture. And there is a quiz that covers some of that content. So please do read that extra stuff about tension and recovery, essentially, with muscles. But we have a lot that we're covering today on its own. So all of our muscle tissue, whether it's skeletal, smooth, or cardiac, have these four characteristics in common. All of them have contractility, meaning they can shorten, which is how our muscles perform their action by contracting and shortening, thus pulling the two ends of that muscle together, or in a smooth muscle, kind of contracting the entire organ itself. They all have excitability, meaning they react to electrical impulses or changes in their membrane potential. So we will be sending electrical impulses down our nerves. Those nerves will release a neurotransmitter at the muscle that they are acting on. That neurotransmitter will cause a membrane potential change in the muscle, and that will send an electrical signal or membrane potential all the way down the membrane of the muscle to then cause muscle contraction. We're going to talk about all the steps of that process and what that looks like in this PowerPoint. They all have extensibility, meaning that they can stretch. And you'll see based on the formation and structure of the thin and thick filaments and the tightened filament specifically, how that would allow for that stretching. So these muscles, the organ itself can stretch up to 3 times its resting length. And then lastly, they all have elasticity. So they are naturally drawn to snap back to that resting length. However, this does rely on the presence of ATP, so the presence of that cellular energy, and we'll look at why later as well. So all of our muscle cells, again, have that contractility, excitability, extensibility, and elasticity. So they can shorten, they can stretch, they are naturally drawn back to their resting length, and they react to electrical impulses, to membrane potentials. Our skeletal muscles are our only example of voluntary muscles, meaning that we have some cognitive control over them. So signals from our brain tell those muscles when they should contract and when we should try to relax them, essentially. Some of our skeletal muscles are considered automatic. So we have the ability to cognitively control them, but we don't need to. They will act on their own. So muscles of our eyelids, muscles involved with breathing, like our diaphragm, right? Those muscles are skeletal muscles. So they have all the characteristics of skeletal muscle. We have the ability to control them if we think about it. Right? We can cause the contraction or stop the contraction of our eyelids and diaphragm, but we don't need to, so which makes them automatic. Right? They will do it on their own without us thinking about it. But then our other two examples of muscle tissue, cardiac and smooth, they are both involuntary. We do not have any direct neural control of those muscles. They are acting on their own. And so we already looked a bit about at the structural differences of those three muscle tissues with skeletal, smooth, and cardiac, but we'll review that again here. So skeletal muscles are huge. They're very large. They are the longest cells in the body. I don't understand necessarily the qualification that makes an ovum larger than a skeletal muscle, because in overall like surface area and volume, I think a skeletal muscle cell is or a lot of our skeletal muscle cells are much larger than the overall surface area and volume of one ova. But ova are considered the largest cells in the body. Skeletal muscles are the longest cells in the body. They're very, very long. They are multinucleated cells. So the only other example we've talked about of a multinucleated cell in the human body are osteoclasts. There's really only one other example that I know of, which is in our blood, the hepatocytes. So we have multiple nuclei, which just like with osteoclasts, that means they came from multiple stem cells bonding together, kind of merging together. And so all of those internal components, including the nuclei, merge together as well. So we get this one really long multinucleate cell with all the organelles that we associate with eukaryotic cells in it. And these skeletal muscles are organized for simultaneous contraction of that entire organ. So one muscle, each muscle is its own individual organ and will have the same general structure of the tissue types in it as each other muscle. So they are all one organ. And so each of those muscles, each individual organ is set up to contract simultaneously. All the muscle cells that make up that organ from tendon to tendon are set up to contract at the same time. Smooth muscle has one single nuclei per muscle cell. They do not have any of the striations that we associate with skeletal muscles. The myofibrils, which are the contractile components within our muscle cells, are scattered and in different directions. So for most of our smooth muscles, when they contract, they're kind of shortening or pulling in the surface of all parts of that muscle and almost like a circular range, whereas skeletal muscles are linear, again, from tendon to tendon, trying to just pull in one direction. And they are being regulated through a pacemaker type capacity, right? We have regulated signals coming from our brain, not cognitively controlled, but regulated signals on a set pace that contract those types of muscles. And then cardiac muscles similarly have pacemaker capacity, so they're set to contract at particular times on that pace, but their contraction is in a wave, which serves the function of the heart, right? We only find cardiac muscle in the heart. And this wave of contractions is set up to push through each of the 4 chambers of our hearts, or each of the 6 chambers in like a bird heart, etc. Right? They go off in a wave at that set pace to push blood through each chamber, eventually pushing it out through the aorta. And so for most of what we're looking at in this chapter is going to be focused on skeletal muscle, their structure and function. So how they're set up, how they respond to signaling, how the actual contraction within those muscles works. And then we'll talk a little bit about how those same systems look in a smooth and a cardiac muscle. But most of what we're talking about is related directly to skeletal muscle as our main focus. And so most of our skeletal muscles, or I guess, I don't know if I can say that, but skeletal muscle typically connects to the skeleton, hence the name. And if it is going to move our skeleton or have the ability to cause movement of the skeleton, it must cross a joint. So we do have skeletal muscles that connect bone to bone, but do not cross a joint. And so when they contract, they may move other tissues, such as a lot of the muscles in our face, which do not cross a joint. When we contract them, we can still move the tissue in that space, but the bones aren't moving. But those that do move, we have an origin and an insertion. We refer to each of their attachment sites, each end, as an origin or an insertion, which we've been talking a lot about in lab. Right? The origin is typically the stable attachment and is usually more proximal. And then the insertion is typically the mobile attachment, and it is also typically more distal. However, that does not apply to every single case. Some of our muscles, like with our trapezius, when we were talking about the trapezius, depending on which part of the skeleton is being kept stable, the other attachment site is going to cause movement. So the trapezius can keep the axial or the major portion of our body stable, right? The shoulders and vertebral column stable, but unilateral contraction can cause our heads to go like this because we have attachment sites up here. However, we could also keep our head stable and then contraction of those traps can pull our shoulders up, right? So this does not apply to every single case, but in most of them, the origin is stable and the insertion is mobile. So it'll be, when this muscle contracts, it'll be pulling whatever bone it is inserted into towards the point of origin. So our skeletal muscles perform other functions as well, of course, like maintaining posture in anatomical position. All of your joints except for your ankle joints are at extension, and it takes muscles to hold them that way. Even sitting in a chair like this, I have active muscles keeping that posture. So unless you are completely fully relaxed, which is very hard to do while awake, even if you're laying down and consciously trying to fully relax every muscle in your body, it's very hard to do. We are typically having some form of action, of contraction to maintain our body position. So beyond movement and posture, they provide support and protection. They play a role in our blood sugar regulation. We basically only store sugar as glycogen in either our liver or our skeletal muscle. And so if we have high blood sugar and we want to remove that glucose from our blood, our skeletal muscles and our liver are going to be stimulated to absorb glucose from the blood and convert it into glycogen. And then the opposite is true. If we have low blood sugar and we need more glucose available in the blood, our skeletal muscles and liver will be stimulated to break down that glycogen to add more glucose, available glucose to the circulatory system. Some of our skeletal muscles play a role as sphincters, regulating passage from one organ to another. So the structure of a sphincter has smooth muscle lining the internal component and then skeletal muscle lining the external component. And then lastly, skeletal muscle plays a big role in our homeostasis of heat, or at least body heat in general, even if it isn't directed homeostasis. Right? Any kind of work chemical reaction is going to release or lose some energy as heat. And since we have a large amount of chemical reactions required to move a muscle and a lot of energy production, a lot of metabolic reactions occurring in our muscles, there's a lot of heat being produced in those organs as well. Then our cardiac muscles and smooth muscles have much shorter lists of their functions. Our cardiac muscle, again, only found in the heart, is there to pump blood through the heart and into the arteries. Our smooth muscle is lining all of the cavities of organs. So the lumens are those cavities. It is maintaining and changing diameters of our blood vessels and airways, all of those tubes. Smooth muscle regulates the elevation of hair follicles, right? And then plays a role in moving the contents of those lumens through that tract. So any organ system that is a long set of tubes is going to have smooth muscle around there controlling that. So the cardiovascular system, pumping blood through all those tubes, digestive system, moving food through all of those interconnected tubes, urinary system, reproductive system. You'll find smooth muscle in all of those places. Okay. So let's get into the structure of skeletal muscles. So like I said, each individual muscle, which we have a name for, right, is essentially one organ and will follow this same general structure. We have bundles of fibers within bundles of fibers within bundles of fibers. So at the organ level, the largest version here, we have connective tissue completely surrounding the entire organ that we call our epimysium. So some patterns to point out here. We've introduced these prefixes a few times, epi meaning outside of, peri between, endo within. So these are all referring to layers of connective tissue at those levels. So the outer portion, in between, and then on the inner portion. Mesium or mes, myo, as well as sarco are all prefixes that are referring to muscle. So anything with that myo prefix, or if you see myo show up in a word that's referring to muscle, if you see sarco show up in a word that is referring to muscle. So these are referring to our three different layers of connective tissue within each muscle, within each of these organs. So we have epimysium surrounding the entire outer portion of the muscle. Within the epimysium, we have these bundles called fascicles. Each fascicle is surrounded by the perimysium and is going to have more bundles of individual muscle cells. This is where we're actually getting to the cellular level. So you can see a bunch of different words referring to one muscle cell. A muscle fiber, a myofiber, or muscle cells are all describing one single muscle cell. And so a fascicle is a group of multiple muscle cells, and that fascicle is surrounded by perimysium. And then at the cellular level, each individual muscle fiber or muscle cell is surrounded by endomysium. So again, epi meaning outer. This is connective tissue surrounding the entire outer portion of the organ. Peri meaning between. These are surrounding each of the fascicles, which are groups of myofibers of muscle cells. And then endomysium surrounding our smallest unit here. Each individual cell is surrounded by endomysium connective tissue. And then even within those muscle cells, we have more bundles of fibers called myofibrils. And this is where we're going to be spending a lot of time looking at today and focusing on. Right? So when we're talking about how our muscles contract and the functions they're performing, that is at the myofibril level. So once we are signaled, once a neuron brings a signal to stimulate contraction, a lot of what is happening is going to be at the myofibril level within the muscle cell. Okay, so these myofibrils are more bundles of fibers, and then we'll talk about the sarcomeres, which is one contractile unit of a myofibril. So we got a few different levels of organization here to consider. This is talking about the same stuff with a slightly different figure or a slightly different view. Right? So you can see that epimysium around the outer portion of the entire organ, the perimysium kind of penetrating in between, surrounding each of our fascicles, and then we have endomysium surrounding one individual cell within there. Yeah, I believe that's all I wanted to talk about here. So now looking at the cellular level. So again, myo, a prefix referring to cells, and also sarco, another prefix referring to muscle cells or muscles. So each muscle cell, although it looks a lot different than any of the other cells that we typically talk about or show images of, right? So these very long cells, again, multiple nuclei on the most outer portion, but they do have a membrane, they do have an endoplasmic reticulum, right? Of course, they have nuclei. And so we have different names for those structures specific to muscle. So the sarcolemma, referring to the membrane of muscle cells, the sarcoplasm, referring to the cytoplasm of muscle cells, the sarcoplasmic reticulum, referring to the endoplasmic reticulum of muscle cells. Okay. They've all been given their own names for this. So the membrane of the muscle cell, the sarcolemma, has a lot of those invaginations or penetrations into the cell itself. So penetrating in between the different myofibrils. And that's going to be important for that signal transduction. So in order for the membrane potential carrying that electrical signal to the muscle cell to start contraction, that is going to be carried along the membrane through these invaginations, these transverse tubules or T-tubules. These are just invaginations of the sarcolemma of the membrane in between myofibrils. And so that'll typically come in between two different sarcoplasmic reticulums, which are holding these storage units full of calcium, which is going to be what activates the contraction of the muscle, essentially. And so those storage units in the sarcoplasmic reticulum are called terminal cisterns. And we're going to take a look at images of these things. So I'll talk about them again when we get there. But so essentially the sarcolemma penetrates down to in between two sarcoplasmic reticulums, specifically at their terminal cisterns. And so we refer to one of these T-tubules with the two neighboring terminal cisterns as a triad. Okay. Again, sarcoplasm, just referring to the cytoplasm of the muscle cell. We have multiple nuclei because it's multinucleated. We have many, many mitochondria, and the number of mitochondria will differ between like fast muscle fibers and slow muscle fibers. We have myoglobin. So you're probably familiar with hemoglobin, which is the pigmented protein responsible for carrying oxygen in our blood. muscle cells have their own version of that protein, myoglobin, to carry oxygen and essentially store it to help with ATP production. Okay, I'm going to make a few references to our cell biology PowerPoint here as we're talking about the use of ATP and energy in our muscles to remind you of some of the stuff we learned about cellular respiration. And can I get another quick confirmation? Did this accurately flip to the other PowerPoint, or are you still looking at the muscles right now? It flipped. Okay. Thanks, Constance. Cool. So yeah, I'm going to be jumping back and forth here a couple times in this PowerPoint to talk about cellular respiration, but muscles having their own protein to hold on to oxygen and store that for them is really critical for allowing the full process of cellular respiration to occur. So to get our most efficient production of ATP, we require oxygen to draw that pyruvate into the mitochondria. And when we start running out of oxygen during extreme exercise or extreme intensity or higher intensity anyways, we don't get to do those last two steps of cellular respiration. And we can only do glycolysis, which is much less efficient, produces pyruvic acid as a, or not pyruvic acid, lactic acid as a byproduct, and that comes with other issues. So we'll come back to that later, but this myoglobin is super important in that function. It is storing oxygen to assist with ATP production. And then again, the actual contractile unit within these muscle cells are the myofibrils. And each of the contractile portions of that myofibril are organized as sarcomeres. So we're going to look closer at the structure of sarcomeres as well. All right. So just this figure is just showing you the production, the formation of an individual skeletal muscle cell. So just like with osteoclasts, we have multiple stem cells, essentially, that are growing in size and then fusing together to make one cell, right? The osteoclast is really large. It is multinucleate because multiple cells came together to make it. Skeletal muscle is generated in a very similar form. We have multiple stem cells that then fuse together. So add all their cytoplasm and organelles together. And so we get one large, long cell that has multiple nuclei in it. Our muscles also have these myosatellite cells, which are there to regulate them, right? Help assist them in their formation and function and regeneration, these myosatellites. But a majority of the mass is that one muscle cell itself with its many bundles of myofibrils. Okay, and here's a good look or a closer look at the striations. So these lines that are characteristic of skeletal muscle and cardiac muscle, these striations, these are formed based on the location and organization of the actual protein filaments that make the contractile units of these muscles. So each of these lines that you see here, sorry, kind of skipping ahead a bit, is this inner portion of the sarcomere. Okay, so we get each of these lines, these dark, dense groupings and organizations of these myosin fibers creates that striation. Okay. So this is arguably the most important figure from this chapter as far as building an understanding of muscle function. We get to see all of those invaginations of the sarcolemma, creating those T-tubules, which we see in yellow here. These T-tubules are in between two different sarcoplasmic reticulums. And so each of the ends of the sarcoplasmic reticulum here is called a terminal cistern. And in those terminal cisterns, we have a whole bunch of calcium ions stored up. And that calcium is what allows the muscle fibers to actually contract. So when a signal from our nerves reaches a muscle cell, that's going to send that signal is going to change the membrane potential, sending a signal down the plasma membrane into these T-tubules and down the membrane of these T-tubules until it reaches the terminal cisterns. That change in membrane potential is going to open up the calcium channels within the membranes of those cisterns, which is going to allow calcium to flood into the myofibrils, and that calcium is going to allow then the myosin heads to cause contraction. Okay? So understanding the organization of the muscle cell from this viewpoint is definitely something you will see in an assessment, right? This is arguably, again, the most important figure from this textbook showing how each of these structures is organized in order to give an understanding of then how it functions. And so you can see we've got lots of mitochondria in here as well, again, to help with that generation of ATP to power all these different chemical reactions that are occurring. Trying to see if I want to, yeah, let's do a check-in really quick. I know so far it's been a lot of info dump with new terms and structures of things. I want to get a quick fist of 5 on your confidence with the organization of a muscle, these different levels of structure. Still waiting on 4 of you guys to answer, but some people may have walked away from their computer. So I'll take this as the data I needed. So mostly 3s, a few 2s, and a 1-4. So I guess that kind of lines up with what I would have expected, right? Because it's a lot of terms, it's a lot of fact so far, right? I haven't really addressed many concepts besides the bundles within bundles. So not feeling confident enough to answer a short answer question or to teach it to someone else at this point is totally expected. But do you guys have questions that I could answer to maybe help with this? Was any of this confusing, or it's just a lot of info to take in?
I think it's just a lot, even with having kind of scanned over the material beforehand. There's just a lot.
Okay. So again, yeah, the concept here, or the only real conceptual understanding, is just we've got bundles of fibers within bundles of fibers, right? And there's something separating each of them. But most of the rest is terminology and facts. So we stopped off looking at one muscle cell, and now we're going to look closer at the structure of the actual myofibrils. So these myofibrils are just organizations of protein filaments. And so we really have separation into 2 types of filaments that are made up of the same type of protein. So we have our thick filaments, which are made up of myosin. And then we have our thin filaments, which are made up of actin. So essentially all the parts that you see in purple here, which are made up of myosin, it's that thick filament, the location of these thick filaments is not going to change during contraction. How these muscles actually contract and shorten is that the heads on these purple lines, on these thick filaments, are going to pull on the actin on our thin filaments, bringing them closer to this M line in the center. So this purple part will basically stay where it's located. There's a bunch of little tiny like arms essentially coming off of these thick filaments that is going to grab on to the actin and pull them in one direction. So on each side, we have arms pulling towards the center. And so we're going to pull these thin filaments towards the M line, the center of our sarcomere here. And so that looks like this. When I was learning about muscles, we were asked to memorize the different names, the I band, the Z line, the A band, the H zone, all those things. I have never ever had that come up in my life since then, and thought it seemed tedious and unnecessary at the time. So I'm not going to assess you on these different names. They're just here for your knowledge. So I would rather, or I only care about understanding the concept. The thick bands are myosin in the center here. These do not move. They are going to attach to the actin and pull the actin towards the center. So on either side, these heads are pulling the actin filaments towards the center. And so our thin filaments are made up of multiple different types of actin. And the main ones that you should be aware of and that we'll be talking about a lot or referring back to as we talk about how muscles contract are tropomyosin and troponin. I guess as well as the G actin that we have in here. So let's just look at this figure. Right? So we have a bunch of these little red and yellow balls here. These are each G-actin filaments, these little bundles of G-actin. And the yellow that's being covered by this green rope-like protein, that yellow portion of the G-actin is the active site. And so in order for our muscles to contract, these myosin heads of our thick filaments, all these little things that you see branching off here, these myosin heads need to be able to bond with that active site. So at rest, when our muscles are not being stimulated to contract, all of those active sites are being covered by this rope-like protein called tropomyosin. That tropomyosin is being regulated by this smaller brown protein that we show here called troponin. And again, these are all just different types of actin. So we have our G-actin, which is where the myosin is going to attach. We have tropomyosin covering those active sites when our muscles are at rest. And then we have troponin, which is basically regulating the position of that tropomyosin. So a concept we haven't explicitly addressed, but has kind of come up and leaked into this class in multiple different ways, is that proteins will almost always change shape when they bond with something. So majority of protein function has to do with which kinds of things it can bond with. So its shape and its chemical composition is going to be specific to very particular things, like calcium in this case. So the shape of the protein is specific to what it can bond with. When it does bond with something else, it typically is going to change shape. And that change in shape is what is going to then perform the actual function. So in this case, we have tropomyosin covering the active sites of these globular actins, these G-actins, and troponin is regulating that position. And when it bonds with calcium, it is going to move, change shape, and that is going to move tropomyosin off of all these heads. So when calcium floods into the muscle cell, as we're going to talk more in depth with neuromuscular junctions and how our muscles actually contract. When calcium floods into these myofibrils, it is going to bond with these troponins, actins, these troponin fibers, and troponin is going to move tropomyosin off of the active site, thus exposing it to the myosin heads. Okay. So like we have other portions, other types of actin to talk about, like there's titin, the nebulin, etc. Those, for the purposes of understanding this concept, you can kind of ignore. Right? Nebulin is just that central protein fiber that all these globular actins are attached to. Titin is the version of actin that is connecting each of our strands to another border or another fiber. But as far as how this myofibril is going to act, we care about troponin, tropomyosin, and then the globular actin where the myosin is attaching. So again, calcium floods in, it bonds with troponin, troponin changes shape, which is going to move tropomyosin away, and that allows for myosin heads to now bond with the active site of G-actin. And so these myosin filaments are what make up the thick lines. So that was all actin of these thin red lines. Now we're looking at myosin, which makes up the thick filaments. Myosin is a different type of protein. We have these heads that branch off, and their whole job is to basically hinge, very similar to a joint. Right? Basically, in the presence of ADP with its own phosphate, and then the absence of those things, and the presence of ATP again, that is going to move the position of this protein as it's bonding and releasing them, which allows for what we call a power stroke. So calcium flooded in, it bonded with troponin. Troponin moved tropomyosin off of the active site. Now this myosin head can attach to these globular actin balls here. So that head will attach. It will then release ADP and an individual phosphate, which is going to cause that power stroke, pushing or pulling those thin filaments closer towards the center here. And then it will release it when a new ATP molecule bonds with it. So that is all one motion. We have lots of these myosin heads all acting at the same time in lots of different myofibrils, or all of the myofibrils in one myofiber. And we've got multiple myofibers that could potentially be activated at once. So we've got a lot of movement happening at once to do this. Okay. Um, and when I was learning about muscles, it was the first time that like relativity clicked for me. Learning all the steps that are occurring as you think that I want to move this muscle that is moving a bunch of ions in and out of your neurons, which is causing signal transduction through millions of neurons all the way down your vertebral column to whatever muscle you're going to move. That is releasing neurotransmitters that cause more signal transductions to move down the membrane of your muscle, which is going to cause the release of calcium, and that calcium needs to bond with troponin, and the troponin is going to move tropomyosin. And then myosin needs to bond with that actin, release ADP and PI to pull that muscle. And all that is happening in an instant every single time you move a muscle. And so realizing like how much stuff is happening in a unit of time that I can't even comprehend or measure was mind-blowing for me at the time. So maybe, hopefully, that is getting across for some of you too with some of this stuff. Okay. So now let's talk more about that process of different steps involved in getting a muscle to actually fully contract. Okay, so getting into a position where a myosin head can bond with those globular actins and pull your muscles to shorten them, to contract them. So every set of muscle fibers that are going to contract at the same time are all being controlled by one neuron, essentially. So a motor neuron is a neuron that is innervating with that muscle fiber. And so where the axon of that neuron meets the muscle fiber is called the motor end plate. And they're typically like branching in a lot of directions, so covering a pretty significant surface area at that axon terminal for this motor end plate. And so that signal is going to act on this entire muscle cell, sending that electrical stimulus all the way down the sarcolemma here. And so together, where a muscle meets a neuron is referred to as the neuromuscular junction. Okay, so the motor end plate is the portion of the sarcolemma where that axon terminal extends, and then where a neuron is meeting the muscle fiber is the neuromuscular junction. And so we'll look at all this stuff in more depth as we go. And so we're going to be talking about contraction from the perspective of each motor unit. So one single motor unit is describing all of the myofibers, so all of the muscle cells that are going to contract in response to one motor neuron. So a motor unit are all of the muscle cells that will react to one motor neuron. And so some of our motor units are smaller, meaning that there are less muscles being innervated by that one neuron, which allows for a lot more control. So like our tactile movements of my fingers, which needs to be a lot more precise as I interact with my environment. Those are smaller motor units compared to the motor units of like my biceps or my quadriceps, right, which have to do very large movements, moving a lot of muscle cells at once. So those are much larger motor units. And basically, the larger the motor unit is, the more force that muscle is going to be able to generate. And we can also increase the amount of force being generated with increased frequency of stimulation. So force is generated in specific increments with each time it's stimulated. There's a cap to that. One of the crash course A&P videos that I linked in the module for you guys does a good job really showing that and talking about it. It is discussed in the chapter a bit, like the summation of force. But essentially, we increase force by stimulating multiple muscle cells at once, more muscle cells at once, and by increasing frequency of that stimulation. Muscle cells can also recruit other motor units from that one stimulation. Right? So that's referred to as recruitment. And then the synaptic cleft is just the space between the synapses at the ends of our axon and then the actual sarcolemma. So there is space in between this neuron and the sarcolemma, the membrane of the actual muscle fiber down here. And in that space is where we get to see some of this more molecular movement. Trying to remember yeah, I think this is just here for reference. We're going to talk about all the specifics step by step here. So as a signal travels from your brain to this muscle and reaches the neuromuscular junction, right? All these signals are electrical. They are based on membrane potential. And I'm realizing maybe I should have talked about that first with how this is organized. Yeah, I am going to kind of jump around here for a second. So what we're about to talk about, or yeah, what we're about to get into, we're going to have a lot of reference to membrane potential. And membrane potential is essentially describing the difference in charge that is inside the cell versus what is outside of the cell. So as ions are moving into or out of the cell, that is going to change the charge of both environments. So in this case, we're looking at the sodium-potassium pump, which is always active in almost all of our cells, basically regulating the general charge of the external and internal environment of our cell. So the sodium-potassium pump moves 3 sodiums out and 2 potassium in for every ATP it uses. And so we are losing 3 positive charges and gaining 2 positive charges. So a net loss of 1 positive or a net gain of 1 negative, however you want to look at that. Essentially, the charge is going down by 1, or the internal charge of the cell is going down by 1 every time this sodium-potassium pump uses some energy. And so we can regulate the charge of this environment through this pumping mechanism. Again, every time this action occurs, the overall charge of the cytoplasm is going down by 1. So whenever there is a directed signal being sent from a neuron, or in this case, being sent down the sarcolemma of our muscle, what's happening is instead of a pump like this, which is regularly moving ions in and out, we get channels that are going to open and close, causing very quick, large changes in our membrane potential. So essentially, if a channel like this, or this, this is showing our calcium voltage gated channel. This is showing the sodium channel. So when a signal like acetylcholine, a neurotransmitter here, binds with this sodium channel, that's going to cause the protein to change shape, which in this case is going to open space for a bunch of sodium to flow into the cell. Sodium is an ion, is a cation. It's carrying a positive charge. So every sodium that moves in is increasing the cytoplasm's charge by 1. And essentially, there are different breakpoints that will then activate other channels that will open and cause a change in membrane potential. And so again, it's just this change in charge, manipulating the charge of the internal environment that is going to activate other protein channels to open that allows for all of this to function. So our neurons do this. We're opening a bunch of sodium channels to change membrane potential, which sends that membrane potential down to the next neuron, et cetera, keeps going. And now we're looking at it with our muscles doing it, sending this flooding of changing of membrane potential down the membrane. So hopefully that preface was helpful rather than more confusing, because we are going to now look at the application. So essentially that action potential is that change in charge. It starts causing this chain reaction where the membrane is going to change its charge all the way down as it moves. And this is going to cause these vesicles that are held within the synapses. So at the axons, at the ends of our axons here, at the synapses, we have a bunch of vesicles holding neurotransmitters, which are proteins that are going to cause another reaction. So we have these vesicles, and in this case, carrying acetylcholine. So as this membrane potential reaches the axon terminal, those vesicles will bond with the membrane and release acetylcholine into this space. The acetylcholine will start to bond with these channels, these ligand-gated ion channels. So this will cause them to open, which is what we were seeing here. So acetylcholine bonds with the protein. It causes the protein to change shape, opening this channel. And so now sodium has an area where it can just flood into the cell. that's what we're seeing happening here. Acetylcholine is bonding with those membrane proteins that's causing the channel to open so sodium can flood into the cell, which is going to start changing the membrane potential, and that signal is going to cause a chain reaction that is going to flood in both directions, which is where we're starting to see this red on the membrane show up. So that membrane potential, that action potential, is going to start spreading down the membrane. We then have acetylcholine esterase, a different protein, which is there to basically clean that up. So this is pulling acetylcholine off of those channels, those channel proteins, so that they will close and no longer allow sodium to flood back in. The sodium-potassium pump will then fix what had been left over, right, to get that membrane potential back to where it was supposed to be. And that action potential is going to continue all the way until we reach the T tubules, or towards the ends of the T tubules. Let me see where that's at. Oh, I need to move backwards. That's fine. Okay. So the action potential came from the neuron, released acetylcholine. Acetylcholine opened sodium channels, and so that then started an action potential on the sarcolemma. So that action potential will travel all the way down the T tubules until they reach those terminal cisterns of the sarcoplasmic reticulum. And again, those terminal cisterns are holding a whole bunch of calcium. And so that membrane potential, as it reaches the membranes of the sarcoplasmic reticulum, is going to then cause calcium-gated channels to open, which is what we are seeing here, right? So typically at resting membrane potential, which is negative 70 millivolts, those channels are closed. As sodium is flooding into the cell and increasing the charge of the cytoplasm, so instead of negative 70, now we've moved up to negative 60, so we've increased the charge by 10 millivolts here. That is going to cause the protein that is blocking that channel to change shape and open. And so now all the calcium that was being held in the sarcoplasmic reticulum is going to flood into the myofibrils until eventually we reach another threshold where those gates will close, so the calcium will stop doing that. But that calcium being released into the myofibrils is really important because that is what is going to move those tropomyosins off of the active site of G actin. So calcium comes out of the sarcoplasmic reticulum. It bonds with troponin. Troponin is going to change shape and move that rope-like tropomyosin off of the active site. That's what you see here, right? Calcium bonded with troponin that moved tropomyosin off of the active site. And that is going to allow for myosin to now form a bond with that active site and then allow it to do its contraction, to do its power stroke, moving our muscles closer together, essentially, these myofibrils. So now looking at it from this direction, right? So this is a resting sarcomere, so not contracted. All the myosin heads are detached from the actin filaments. So this should be basically in this resting form that we see here. So the thin filaments are spread out. They are not close to that central line, and the myosin heads are not attached. Okay. So as calcium comes in, troponin moves to tropomyosin, and now these active sites are available. And so myosin is going to very happily and readily bond with those active sites. That's what we see happening here. It will then release this ADP and individual phosphate. And as it releases something, again, the protein is going to change shape. If it bonds or releases something, it changes shape. So as it releases that ADP and individual phosphate, that's going to cause the power stroke, the bending of that head, that hinge, pulling the thin strands of the muscle closer together. And that is contraction. So we have a whole bunch of myosin heads pulling on all the thin filaments in each of our sarcomeres and each of our myofibrils and each of our myofibers to cause this one big contraction. But now that the stroke is done, the contraction is done, if we want to then relax and rest this muscle and allow it to go back to resting state, we then need to add ATP to bond with that head, causing it to change shape again, which will release actin. So ATP is not only needed to power that power stroke, sorry. So as we release ADP and PI, that is going to power this hinge movement. So ATP is needed for that contraction portion to occur. But we then also need to add ATP back to that myosin head to allow it to let go of that thin filament. And so this is where rigor mortis comes from, like that maintained contraction in a dead or dying piece of tissue or organism. Right? If the body is no longer functioning, you're no longer breathing, delivering oxygen to your cells, which means you're no longer generating ATP, or at least not efficiently. You don't have enough ATP for those myosin heads to release the contraction that they were already doing. That keeps those muscles contracted. They literally cannot let go of each other without that ATP there. Or some other interference to cause them to disassociate. So ATP is required for contraction and relaxation. Okay? So a lot of energy usage going on in the muscle. And then the myosin head will break that ATP apart into ADP and one individual phosphate, which will allow it to snap back into that ready position. But the myosin head releasing from the actin and snapping back does not necessarily mean that your muscle is going to go back to its resting length. In order to do that, there is still some sort of force needed. Right? And so typically this is going to be antagonistic force from another muscle, straightening my arm after I had flexed it, or just gravity pulling on it, allowing it to straighten, right? But it needs some sort of force to then pull those two different filaments apart from each other, or else they will just stay there, right? They're not naturally, there's nothing else pulling them unless we apply some force as well. And so altogether, that is how our muscles contract. A action potential from our neurons releases acetylcholine, or I guess that's all summarized here. So I don't need to talk about it on my own. I can have a figure. So an action potential reaches the synapses of the axon, which is going to cause these vesicles carrying acetylcholine to release that acetylcholine. Acetylcholine is going to bond with these sodium channels, which will cause them to open, allowing sodium to flood into the cell. That causes a chain reaction of this membrane potential traveling down the sarcolemma into the T tubules, where it will then stimulate those calcium channels to open. So calcium will leave the sarcoplasmic reticulum. It's going to move the tropomyosin out of the way, allowing myosin to bond with the actin. And then myosin is going to perform that power stroke, pulling those thin filaments towards the center. And thus we get muscle contraction. And so once contraction is done, right, ATP releases that myosin head. The calcium is going to be reabsorbed back in the sarcoplasmic reticulum. So the troponin is going to let go of it. It'll be reabsorbed back into the sarcoplasmic reticulum. The sodium potassium pump is working to get our membrane potential back to that -70. Right? So pumping sodium out and pumping some potassium back in. And acetylcholine esterase is cleaning up all that acetylcholine, which stops the signal in the first place. So basically everything that was done to cause this contraction is then essentially reversed when we are relaxing that contracted muscle. So instead of calcium being released, it's being absorbed. Instead of myosin attaching, it is detaching. Instead of these channels opening to allow ions to flood in, they are closed, and we are now pumping most of our ions back out. Okay. So for these processes, right, please watch the videos that I've suggested as well. Like with most cellular processes, it's just much more helpful to be able to see these steps in an animation, see them happen thing by thing, rather than looking at these two-dimensional images, trying to talk about what has happened with each thing. So please spend some time like reviewing and watching that stuff to try to generate that understanding. Of course, if you have questions that I can answer about this to try to make it more clear, please ask them. So is there anything about this that I could clarify? So again, there's a lot of steps A lot of things happening at that molecular level in these cells. And so again, imagining like how many muscle cells there are in just one muscle, one large muscle like a bicep or your quads, right? All those four muscles that make up your quads. Right? So much of this is happening in just an instant, right? That we can't even comprehend the time or the level that these things are they're acting on. So much movement of ions and movement of proteins to cause that muscle to contract. Okay. So we're going to wrap up here with a few slides talking about energy regulation in our muscle cells, a look at the difference between fast and slow fibers and what that really means, and then a quick look at what this contractile unit looks like in a cardiac and a smooth muscle. I think we just have 2 slides for each of those things. So in our muscles, like with most of our cells, we need ATP to power any kind of function. So ATP or energy is being utilized for all chemical reactions. And so we have a lot of those really large movements occurring. We need a lot of ATP available for our muscles to perform their function. And so we can't really store much ATP. Each of your muscles has enough ATP floating around to power essentially 2 seconds of movement, of contraction. After that, we have a unique phosphate carrying molecule that isn't really present in any of our other tissues or cell types called creatine phosphate. Creatine is basically a very tiny protein made up of a few amino acids together that is just holding phosphate to help replenish ADP into ATP again. So it's just like our quick backup to make some new ATP for just a few more seconds of contraction of power. So the next 15 seconds of that muscle movement, of that contraction, is then going to be powered by creatine phosphate. That's what you see being shown here. Okay, so ATP is being broken into ADP in order to provide power, and we only have enough to power about 2 seconds of contraction. Creatine phosphate is then going to be used to quickly replenish some of that ATP, so giving that phosphate to ADP, making some new ATP, so we get 15 more seconds of contraction from that. And then we are generating ATP through cellular respiration, okay? Through those 3 processes of cellular respiration: glycolysis, the citric acid cycle, and then the electron transport chain. And the substrate that we start with in cellular respiration is glucose. And so muscle cells have a lot of stored glucose in that storage form, that polymer, that polysaccharide called glycogen. So we have a bunch of these individual glucose molecules that we can just pop off of that stored glycogen in order to do cellular respiration. So there's a lot of energy available in our muscles, and where the energy is coming from is just going to change depending on how long that contraction is going to last and at what intensity, how much force we need to generate. So in a resting muscle, rather than taking new glucose from the blood to try to replenish this glycogen and stuff, we're going to be breaking down fat, mainly fat. Right, so we have that ATP for those first couple of seconds. We got creatine phosphate to quickly regenerate that ATP, and then the rest is going to be through cellular respiration. In this case, with a resting muscle that does not need that ATP extremely quickly, all of the cells in our body go to fat as the energy source when we have enough time to break it down. Fat is much more energy dense. It has 9 kilocalories per gram compared to 4 kilocalories per gram in carbohydrates. So when we have the time to fully convert a fatty acid into something like acetyl-CoA or pyruvate, we will do that first. So at a resting muscle, once we've run out of ATP and run out of creatine phosphate, we're mainly going to use fatty acids as the substrate to then go through the process of cellular respiration. We have myoglobin in our muscles, which is holding oxygen to help power the processes that occur in the mitochondria. So we should have plenty of energy sources when we are at rest for our muscles to generate the ATP they need without any other noticeable side effects or issues. As the intensity of the activity is increasing, we're going to need that ATP to be produced more quickly. And so although fatty acids are more energy dense, they are slower to break down. They require more steps. And so we're going to start switching more and more to using glycogen, or stored glucose, or stored carbohydrates, as the energy source. And again, at like moderate activity, we should have enough oxygen from both myoglobin and from the oxygen that's traveling in our blood from our lungs to power the cellular respiration processes needed to generate the ATP that we require. So the only real difference here is that we're starting to take oxygen from the blood as we're increasing activity level, and we're starting to look more towards carbohydrates as the energy source instead of taking the time to break down fatty acids. But then as we get into intense activity or towards the peak, the highest level of energy usage that our muscles could do, we are not going to be able to keep up with the amount of oxygen needed for cellular respiration to happen fast enough. Right? These muscles use a lot of ATP to contract and to generate force. And so if we're doing that at a high level, that ATP is going to be used much faster than we can break these other molecules apart to make ATP. And we're going to use up that oxygen way faster than our circulatory system can get it to our muscles. And so when we run out of oxygen, we can no longer do all three phases of cellular respiration. And instead, we're now stuck with glycolysis as our only source of ATP. And there are multiple issues that come with that lack of oxygen. Right? So if we're only doing glycolysis, we're making much less ATP per glucose, right? We have to put 2 ATP in to begin with, and then we get 4 out. So we're only making 2 ATP per glucose molecule through glycolysis instead of the 30 to 36 that we can get per glucose molecule if we have oxygen present. So one of the issues is we're generating much less energy. The second issue is that if we're not going to perform the TCA cycle and the electron transport chain, then this electron carrier has no use to the cell. And so since we have this molecule carrying an electron and it has nowhere to give that electron to, we need to be able to bring it back to its original precursor. So we need to get this NADH back to an NAD molecule. And so we have to give that electron to something else. And so since this pyruvate isn't going to go into the mitochondria, these electron carriers give their electron to pyruvate instead. When there's no oxygen present, that is what creates lactate or lactic acid, because it's going to release hydrogen ions as it's carrying that electron. And so that's going to increase the acidity of our muscles, just talked about in here. So here we go. The pyruvate is converted to lactate. Hydrogen ions from that are not going to be absorbed into the mitochondria, and that's going to cause the buildup of acidity of those hydrogen ions in our muscles, and that inhibits the proteins that are responsible for our muscle contraction. Right? Proteins have very specific conditions in which they can act in, whether that's temperature or pH. And so if the pH is changing significantly, like it does with pyruvic acid, that's going to stop its ability to contract and relax regularly. And that's how we end up cramping. Right? We have excess contraction that we can't control, or we can't control the entire process of neural stimulation and contraction. And so our muscles contract in a painful way because that's not being controlled. That sends a pain signal to us, etc. There's damage, right? So that acidity is what is causing it. And so not only did we increase the acidity here and potentially damage the proteins of our muscles and cause ourselves to feel pain, but that lactic acid, that modified pyruvate carrying those ions, that lactic acid is then going to travel all the way to the liver in order to be converted back to glucose. And converting lactate back to glucose requires energy. So overall, the net production in our body of energy gets canceled out when we don't have enough oxygen in our muscles. So at peak activity, we're using ATP too quickly, using oxygen too quickly. And so we build up this acidic lactate. We then have to recycle that lactate in the liver, which is going to use energy. So the net production of energy gets canceled out, essentially, if you don't have oxygen present. Are there any questions or concerns I can answer about that? Hopefully a majority of this concept is just review of cellular respiration. Okay. one slide here to quickly talk about the difference between a fast and a slow muscle fiber, or a fast twitch and slow twitch muscle fiber. So essentially, for most of our very large muscles doing really large movements, they are what we refer to as slow fibers or slow twitch muscle fibers. They are set up to be more efficient at doing multiple contractions over time and thus reducing fatigue, resistant to that buildup of acid, trying to prevent that. And so for our big muscles, that would structurally look like this. And they end up coming out much darker because of all the myoglobin. It is a pigmented protein. So when it's carrying oxygen, it ends up looking red. It's reflecting red light. And so looks red. And then fast twitch muscle fibers are much more easily fatigued. They don't have myoglobin there to help with that ATP production. And so if they're being overused, they can easily build up that acidity. But these are usually found in much smaller muscles that aren't going to do large repeated movements like the slow muscle fibers are. So like in our example here, we have the muscles of the eye, right? They're very, very tiny. And so unless you are consistently doing, like moving your eye back and forth or up and down for any of these rectus muscles, they are not going to be utilizing a whole lot of oxygen to build up that fatigue. Whereas these larger ones are more likely to get fatigued due to the large amount of movement that they are doing. And so they are structurally set up to resist that. They have myoglobin. We have a lot more invaginations so that we can get supplies from the blood and be innervated by neurons, etc. Yeah. And so we could see this with like the meat that we eat. Okay, so if you think about like a chicken breast versus a duck breast, and the functions that those muscles perform for those two different types of birds. Okay? Chickens can't really fly for extended periods of time. Okay? And the chicken breast looks white or clear with a very pink tinge, with a little pink tinge in it. Because chicken breasts, their pectoral muscles are mostly made up of fast fibers. They are not structured to facilitate a lot of movements over long periods of time. They are structured to do fast movements in a very short burst to allow that chicken to escape a predator quickly, get into a tree branch. But the chicken didn't evolve to fly as a main form of travel. So their breast meat ends up looking white with that slight pink tinge because it's mostly these fast fibers. Comparing that to a duck breast, where ducks are using flight as a main form of travel and they are migrating over large distances. So a duck breast is set up with mainly slow fibers to facilitate that use of the muscle for a long period of time, multiple contractions, and trying to produce or resist fatigue for that. So most of the dark meat that we are consuming is slow fibers, and most of the white meat would be fast fibers. Right? So the legs and wings of a chicken are dark meat, right? Because they have the slow fibers, and then the breast is white meat. It has those fast fibers. So I thought was kind of cool.
Is one considered better to eat than the other?
Sorry, could you repeat that?
Is one considered like healthier to eat or anything like that compared to the other one?
The actual components of like the macronutrients that you would find in a fast versus a slow shouldn't be significantly different, or micronutrients even either. So I don't think it has a lot of nutritional application in that way. It's much more significant to consider how those things are raised and where they're coming from. And if you're getting a chicken from a CAFO versus a wild duck, that is what is going to make the duck breast healthier compared to the chicken breast. But I don't think the actual composition of the type of fiber plays much of a role in its nutritional value. But that would be interesting to learn more about. I can look that up.
Cool. Thank you.
Of course. Good question. Okay. So now, taking these things we've learned and trying to apply that to the slight differences that we see in the structure of cardiac muscle and smooth muscle. So cardiac muscle is much closer to skeletal muscle than smooth muscle would be. Right? We do still have those striations because we have sarcomeres still present, creating those contractile units. Right? So we're going to have our thin filaments and our thick filaments in here. However, the actual muscle cells themselves are smaller, not as long. They're not multinucleate. It's one stem cell that developed into this mature cardiac muscle cell. And then each muscle cell is connected to a neighboring muscle cell through these intercalated discs, which was a really fancy name that they decided to utilize for gap junctions, basically. So that this protein connection that holds those muscle cells together, that allows the movement of ions in between them. Right, so rather than having one long muscle cell, we have multiple muscle cells attached to each other through these gap junctions that we call intercalated discs. And the benefit of that is that it allows some branching and getting some different directionality to the muscles in our heart, okay? Which allows for the shape of the heart and for the pumping of chamber to chamber. Other than that, the general structure of these muscle cells is very, very similar, basically the same. So except there's one nucleus and they're not as long. Other than that, the sarcolemma is invaginating into the actual muscle cell in between each myofibril, reaching the terminal cisterns, and the reaction is the same, right? We've got an action potential that causes the release of calcium, which bonds with troponin, which moves tropomyosin, which allows myosin to bond and power stroke. Right? We just don't control that action. So again, the only differences are they're single nucleated cells. The cells are much shorter, they're not as long, they don't go around the entire heart. And each muscle cell is connected to a neighboring cell through these gap junctions called intercalated discs, which allow the muscle to branch instead of just being one long strand. Smooth muscle, however, is structured quite differently. So similarly, we're going to have myosin acting on actin, so thin and thick filaments, but we do not have those sarcomeres, those contractile units. And the structure of the smooth muscle cell is better set up to squeeze from all directions. So you can see we have these intermediate fibers on the external surface, essentially, of this smooth muscle cell. All of the actin filaments that are going to be pulled by the myosin are attached to those intermediate filaments at these dense bodies. And so when myosin pulls on the actin, instead of shortening a sarcomere, like we saw with skeletal muscles, it's just pulling these dense bodies towards the center of the cell, essentially. And so we get this contracted, squished looking muscle rather than just a shortened version of what we had before. So that's what you see here. So we have intermediate filaments, which typically are structural cytoskeletal filaments. They create like the skeleton that these other protein filaments are acting on. Actin is connected to those intermediate filaments at dense bodies. And then we have myosin kind of scattered that is going to pull on those actin filaments, which pulls all of the intermediate filaments towards the center, creating this squished view of the cell. And so here we have a combined image of a longitudinal scan versus a transverse scan, just two different views of that smooth muscle. You see they don't have nearly the same type of organization and shape that we were looking at with skeletal and cardiac muscles. They're not parallel, but they're also not branched, not really connected directly to each other. We've got connective tissue in between, and so a very different structure. And that concludes the content in this PowerPoint. So I know that was a lot of info, a lot of specifics, a lot of steps as far as the organization